in January and February of this year, and I personally had an encounter with them off the mouth of the Juan de Fuca. That was G-Clan. I was out with one of our operators who departs from Port Renfrew. I was out hoping to see a whole bunch of humpbacks, saw a whole bunch of G-Clan instead. That was my first encounter with northern residents, so you never know what you'll find out there. And then southern residents, of course, this is the type of whale that I certainly get asked the most about, and as I mentioned, it's the type of whale we see least frequently right now among the Pacific Whale Watch Association. Currently 73 individuals. That does include 2 new female calves, so that's very promising, the fact that both of the new calves this year are females. There was a pregnancy reported in June as being a late-stage pregnancy. Haven't heard any updates on that. That whale has been seen in recent weeks, doesn't have a calf with it right now, so I don't have any updates on that. 2 losses this year that we know of. Notably, one of them that I really just want to highlight is K44 Ripple. It's not conclusive, but there is certainly some evidence to support that that whale possibly perished due to entanglement off of Oregon in a crab pot. So often we talk about acoustic disturbance and pollutants and things like that, but entanglement might be something that we really need to talk about more for all types of whales, not just the big baleen whales. And then some whales listed as vulnerable as well. That has to do with body condition. Southern residents, this is where we encountered them. You can see the dots on the map there. I mentioned that this is the type of whale we see least frequently, but they were actually here confirmed 128 days, which is already way more than they were here last year. And we don't have the November numbers out yet, but I can tell you that today is the 19th day of November that they were in the Salish Sea. So they were only here 103 days total in all of 2021. So I'm hearing that there's some pretty good fish returns this year. Hopefully these whales are finding a lot to eat. Presumed presence is a new stat that Orca Behavior Institute started using this year. Basically, if you wake up in the morning and see southern residents on the west side of San Juan Island at 5 in the morning, you can guess that they started swimming in on the day before. So we have presumed presence on days that we didn't have to see them. And then as I mentioned, we're 52 days that we encountered them. If you're not aware, I'll just take this opportunity to let you know we're not allowed to view them if you didn't know that. So in Canada, as well as in Washington, there are very few circumstances that professional whale watchers are allowed to view southern resident killer whales. So if we do see them, we're supposed to report it, but then we depart the scene. So a lot of the dots that you see on this map are actually incidental encounters. And then sea otters. I knew we were going to talk about sea otters today, so I wanted to share with you that we are seeing, I heard all those ohs. Yeah. We are seeing at least a noteworthy increase in sea otters here in the Salish Sea. Now, I'm not talking, hundreds of sea otters or anything like that, but for quite a long time, we only had 1, and that was, if you're familiar with Ollie, who has taken up residence at Race Rocks since 2015, but we're starting to see a few more come in, maybe 6, 7, 8, and they're not just coming in for a day and leaving, they are setting up residency. We have one that's been in Rosario Strait for at least about 3 or 4 months now. There's some that are off of Souk that have been staying there. So again, I'm not going to declare that they're repopulating the area or anything like that, but we're definitely seeing some of them. So it's not as rare as it used to be. And then very, very briefly, I want to share with you some other stuff that we collect information on, which is human actions and human sightings. So a few, I guess it was the beginning of last year, we started collecting this information, sentinel actions we call them. So basically, whale watching is a hot topic here in the Salish Sea, and we wanted to show that we're out there, in addition to educating guests and viewing wildlife, that there's some kind of good that we can do. We can give back, well, if you will, while we're out there. And so we started recording our sentinel actions. Some of these include reactive actions. So if we're out there and we see a boat speeding towards whales, we can flag them down, blast our horn. Proactive, so if there's a ferry coming or cargo ship, we'll call them on the radio to warn them of whales. Marine debris, there's lots of derelict fishing gear and garbage out there, so we'll stop and pick it up. And then other is really anything else, but in most cases, for us, it involves just reporting entangled or injured animals to authorities. And so far this year, we've been able to document more than 1,000 of those, 2/3 of which are vessel related. So stopping vessels that are going too fast, getting too close to whales, giving them a proactive heads up. And I'm very happy to say that in 70 73% of those vessel-related encounters, we have been able to see a visible improvement in that vessel's behavior. So we see them slow down, we see them, divert. If it's a ferry that we're contacting in advance, they'll tell us that they're going to, change course, so we can document that as a positive change in behavior. And that's important because law enforcement is really not out there as often as I think many people realize. And I'm not saying this to criticize law enforcement, and this is also not specific to Canadian law enforcement. This is any enforcement agency on either side of the border, since we do share waterways, but they're only there for about 2% of the time when we are out there with whales. And so a lot of times we do consider ourselves to be that only line of defense. And so I really want to kind of encourage folks to consider that whale watchers can, have a positive impact on the habitat of these whales, which I know is kind of radical, but just hear me out. And then, of course, obviously, whale watchers talking about the good that whale watchers can do. We're a biased source, but there was an independent study that was done and published last month. So using both shore-based observations as well as boat-based observations, Orca Behavior Institute observed the area that you see there in gray, and found that the number of dangerous recreational vessel incidents around killer whales, both big killer whales and southern resident killer whales, more than doubled if there was not a whale watch vessel there. And so this is obviously something that we're very interested in learning more about, but again, I know it's out there, but we're just saying consider the fact that having at least one whale watch boat there to kind of mark the location of whales and model the appropriate distances could actually provide a benefit. All right, thank you.
Yeah, so the question was about porpoises, Dow's porpoise, harbor porpoise, things like that. And where's Anna? She'll be so disappointed I didn't talk about them today. But yes, we do collect that information. We are working with a couple of researchers about large aggregations, so like groups of 50 or more. We do still log it, so it's internally we'll communicate that among our members of where porpoises are. But right now we're really kind of more interested in just the larger aggregations of those.
It may be well known, but water temperature with climate change, does that affect any of these things you discussed?
Yeah, so the question was about water temperature and if that's affecting, what we see out there. I am not even going to pretend to be an expert on that. It's my understanding, and someone here in this room can correct me if I'm wrong, that our water temperatures are still fairly consistent. Of course, there's a warming trend, but I'm being told that, we're not having those wild fluctuations like they did, like with the blob. We didn't feel the effects of that inland as much as on the outer coast, at least is my understanding.
Thanks very much, Aaron. Next up, we've got Fred Denisco and Kendra Nelson as a joint talk.
That would be great. Thank you.
Hello. If you build it, they will come. So, hi, I'm Kendra Nelson. I am the social media coordinator for the Rivershed Society of British Columbia.
And my name is Fred Denisco. I started content creation back in 2021, and both Kendra and I, who have actually connected over social media, have come to see the good impacts and positive impacts that it can have on multiple things, including research and conservation. So we'd just like to highlight some of those. So the first organization that we'd like to highlight is Harbor Wild Watch. Now, I had the pleasure of interacting with Harbor Wild Watch when I first started content creation, and even greater privilege of watching them grow exponentially over time. Now, they are a nonprofit environmental education organization based out of Gig Harbor in Washington, and they have developed a following, especially over COVID, when they started doing digital programming, of over 400,000 followers on TikTok. Now, I don't know if any of you can convince 34,000 people that moon snails are so amazing that you should watch them for 20 minutes, but their last live stream ended up getting 34,000 views for their beach walks. they are also very prevalent with regard to the communities that they've been able to build online, both with the people that are engaging in the local sail or sea area as well as beyond. So they're also very prevalent with regard to communicating with other people and otherwise. One of the biggest benefits with social media that they have seen personally is funding opportunities. So as a small environmental organization out of the Sail or Sea, they were able to secure grants based upon the presences that they had built up over time, which is incredibly great to see. So, yep. When asking their education director about the opportunities that social media provides, the Education Director Rachel Easton responded with, "It's an opportunity to connect with people who may never see the ocean, but nonetheless have an impact on it. We can inspire them to make positive choices and become stewards of their own environment, our own environment, the whole world." So she is an absolute legend. If you do go on to Harbor Wild Watch, please go and like and follow them. They are also looking for verification, so hopefully that might help them out with that. They're also one of the biggest movements that they're going for is tackling misinformation when it comes to online content.
Yes, and misinformation, as we all know, run rampant online. Online, there's some great benefits, but that is a downfall. And another creator that we like to highlight is my friend Emma Luck, who runs a page called Northern Naturalist, and a big thing that she does is talk about marine mammal science specifically, trying to combat misinformation with certain scientific topics, and being there to just answer questions from people, because there are a lot of questions about this field that people have that they don't feel like they can adequately find necessarily either in their high school, at their own university, or from their own community. So being an online resource is really fantastic, and Emma said, oh my gosh, she said, social media is a great tool to reach audiences that may not otherwise have access to science or research, and in my experience, there's a very large group of people out there who are interested in learning more about wildlife. Social media is the most accessible tool for communication with the public, and by utilizing it, we can ensure people are able to access accurate and high-quality information about wildlife and research.
Now, that includes engagement and mobilization. So as a part of a push to try to get the breaching of the Snake River dams onto Biden's infrastructure project in 2021, decided to make a video and trying to get people rallied up to send messages as well as write their names down in support of that. Now, I posted one video casually at this time, gathered 84,000 views, and had a call to action for them to fill out a Google link in my bio. Now, within 3 days, I got 698 messages and 5,006 names pretty immediately, which was absolutely incredible. And so the effects that it can have with regard to engaging people in the kinds of conservation work that we'd like to do, as well as just educating them too on things that are going on, people want to listen. So if you build it, they will come.
And social media can seem kind of daunting. These are big examples. Emma makes beautiful scientific illustration posters for social media. Fred talked about a campaign that he did, but it can be really simple. I started posting on TikTok prior to the pandemic when I was working in my marine science research lab, and I would just post videos of me making salt water, cleaning tanks, feeding fish. People loved it. Some really mundane things that we may not think about. People want to know. Young people especially, they don't really know what's going on in the labs. They think of National Geographic, of Seaspiracy documentaries, things like that about what is marine science, when sometimes it's this. It's a power outage in a lab and making a little funny video about how stressful it was and things like that. And they want to ask questions. They want to learn about ctenophores, which is so amazing for me, that they want to see these things. And that's why building platform and sharing even these little things can be so powerful. Social media can be a big tool in the tool belt of a conservation organization, of a researcher, of a lab, and 40% of Gen Z use platforms like TikTok and Instagram as a search engine, which is why I think it's so important that amazing people like you who are so knowledgeable and involved in this research get online to be that resource for a lot of these youth and a lot of the people, to show them what's going on and just the amazing stuff that you do. So.
Yeah, we believe in you guys, and more than that, we know for a fact that there are so many people out there who are looking to learn about what you have to say. So we hope that this is a good case for you guys to create and build whatever you would like to highlight the things that you're doing. And I promise whatever you're looking for, funding, engagement, opportunities, or otherwise, will come. Thank you.
Thanks very much. Do we have any questions? And I'm just curious, Fred, what is you're producing right now? Do you have a couple of projects in the bag to come out, or?
For myself, I know I would love to get on the water coming into the next summer and go up and down the Sailor Sea. I know there's definitely a few people here that I'd definitely love to talk to as well from me personally.
Kendra?
Yeah, so I work full-time for a nonprofit organization focused on restoring and raising awareness for the Fraser River watershed. So every day I am sitting at my desk making content to raise awareness for that, talking about salmon and all kinds of things. So my day-to-day, I mean, right now it's Giving Tuesday, which is up this next Tuesday. So donate to your favorite organizations and give them money so they can keep doing what they need to do.
Or post about it so they can give you money.
Thanks so much. Thank you. And our next speaker is David Rosen.
Cool. Thanks very much. Thanks everyone for coming. No TikTok videos for me this year, I'm afraid. I'm going to be talking about a very specific project that I did when my research lab at the Vancouver Aquarium was basically hit with this global pandemic thing. You might have heard about it. So a lot of my research program was shut down, and I was scrambling to find stuff. And I got back to sort of the question. I've been working in Pinniped science and management for a long time, and it basically has not changed the question we ask, which is, what are pinnipeds eating? That's decades, that's the same question. The good news is that the technology is changing, and that's what I'm going to talk a little bit about, is an attempt to change the technology to answer the question about the diet and the food intake in this group of animals. And most of our techniques of this is based on the principle of what goes in must come out. And I won't belabor the point of what comes out, but we look into the what comes out traditionally and look at things like hard part analysis, the non-digestible parts of the fish, and say, okay, that's what went in. And we know it's not perfectly accurate. So we've been developing new tools to make it more accurate, including the use of genetic markers in the fecal material that comes out. Very simple principle: the fish or the prey that goes in contains genes. Most of those, or enough of those, pass through into the after food, that we can pick that up, and we can identify the genes from the sea lion or the seal genes, and identify the prey. And this is a technique called DNA metabarcoding. Why it's called that, you don't need to worry about it. And it's really good for identifying prey species that may not show up well in hard parts. And then the thought came, well, okay, if the scat genetic signature is reflecting the food, and if they're eating more of one type of food, should we not see more of that genetic signature in the scat matrix? And that seems like, oh yeah, okay, dead simple, because we know this is the way world works. Let's just go into the field, and some people have done that, and apply that basic principle. Unfortunately, the world doesn't work that simply. There's at least two main problems with this whole thought process. The first problem is what happens of detecting the genes in the scat material. And the other part is what happens when that food passes through a sea lion. So the first part is basically an error in that different fish contain different amounts of DNA, and then even when you take that genetic material and pass it through your magic DNA detection machinery, which is about as technical as I will get, you do not get the same proportion of genetic signature as you do with the genetic signature you put in. That's actually not a big a problem as you might think, because what you can do is basically create fake fish diets in your laboratory by grinding up a bunch of fish. This is a great illustration by Austin Thomas, who did a lot of this work. And you can say, okay, I know what went into this fish mixture. I can say what's the genetic signature, and then I can come up with some correction factors. Easy peasy, not a problem. The problem is number two, literally number two. What happens when the fish passes through an actual pinniped? Because all sorts of interesting things happen. And the only way to really look at how that affects the genetic signature of the scat material is to run some controlled diet studies, which, as you may have guessed, is what we did during the pandemic. We fed a group of stellar sea lions that we have at the Vancouver Aquarium for research purposes some controlled diets made up of coho salmon, capelin, and herring, fed in various known percentages. We fed them over prolonged period of time on each of those diets, collected what came out the other side, sent them to DFO, didn't tell them what the diets were, and they told us what the genetic signature was. And then we got a bunch of genetic back. And this is the only figure I'm going to show you, the only sort of data figure. It's not as complicated as it looks. So on the bottom axis, on the x-axis, is what the diets actually were, showing the different compositions of the different prey, capelin in the blue, salmon in the sort of goldish, and herring in the green. The red bars are the actual diet, so what went in. And along the y-axis, or the vertical axis, is what the genetic material told us the diets were. Now, it may not look that bad. Oh, and by the way, this is not the raw data. If I showed you the results of the raw genetic data, it's horrible. It's nightmarish. But this is with those tissue correction factors applied. So, you know, by eye, it doesn't look that bad, but we're overestimating herring and capelin on average about 20%, and we're underestimating the amount of salmon that they were actually fed by about a third, about 33%, which can be pretty significant when you're trying to apply that to an ecosystem. So you would think, because we had done this before, Austin Thomas had done this, he'd done this with harbor seals, we could just take the harbor seal digestion correcting factors and apply them to sea lions. Bad news, harbor seals are not sea lions, and it actually becomes worse. And this really demonstrates that you have to, at least in large groups of animals, like maybe within otariids, come up with independent digestion corrective factors, because we know the digestive systems of seals and sea lions, although they're both carnivores, they operate in different ways. We think they're related to, again, the type of food they're eating and the amount of food they're eating. And so the question is, okay, we can use this result from cellular sea lions and come up with some digestion corrective factors for these particular set of circumstances, but how universal are they? Well, the next step is try to do that with another set of closely related species. These are some northern fur seals that we also have at the Vancouver Aquarium for research purposes. And so we're hoping to get funding to rerun the same sort of similar study using different prey that are more suited for northern fur seals, such as, you know, some octopus and some squid, and try to determine whether we can come up with more universal correction factors so that we can effectively use these techniques in the field to get a better indication, not just of what they're eating, but how much of different prey items that they're consuming. And I'd just like to finish up with a couple of thanks, particularly Chad Nordstrom, who used to be at UBC here with our group and went to DFO and has really been integral as a collaborator of dealing with all the genetic side. So I just do with the feed and scoop side, and Chad tells me what all the genetic information is. Strawn Tucker, who's been helping work our way through the DFO paperwork, because this original study was paid for by DFO to help support our research program and the Vancouver Aquarium. And finally, I'd just like to thank the continued support of the Vancouver Aquarium for our UBC Pinniped research program within it. Thank you.
Start with Linda and we'll go over.
Mine's silly. I want to know how you got the first in your office chair.
Yeah, it's actually, we were trying out a new veterinarian in lieu of Dr. Martin Helena, and it was working out really well, and then the union came in. No, it's actually, if I can give a semi-serious answer, they're well-trained animals, and this is what I want to emphasize for those who aren't familiar with the Pinniped research program through the Vancouver Aquarium, is we use professional husbandry staff and semi-professional veterinarians to ensure that the animals can do this from a scientific perspective, so we have non-stressed, healthy animals. So really, for them, sitting in Marty's chair is no biggie, but it is really important that they're able to do things like that.
Last question.
You were talking about the correction factors for the DNA. How do those compare to correction factors for your hard parts analysis if you were to do the same thing?
Well, the question was, how do the correction factors for the DNA compare to the hard part analysis? I actually haven't looked at that. We've kept the samples that we collected for genetic analysis to also look at the hard parts to see what it came through. The hard part analysis is difficult for quantification because you have to sort of match up otoliths and ribs and stuff like that. So in theory, what we're hoping is the genetic analysis actually gives us a faster way of quantifying differences in diet as far as proportion of food intake, because it's really a bit of a nightmare to do that through hard parts.
Thanks so much, Dave. And our next speaker will be Kathleen Gill.
Hi, everyone. My name is Kathleen, and I'm a master's student here in the Marine Mammal Research Unit. And today I will be discussing how seals find their way in the ocean. And specifically to do this, I will be analyzing the long-distance movement patterns in southern elephant seals. And while this talk and the talk after it will be looking specifically at the Southern Ocean, I hope if there's one thing that you take away from my talk is that the concepts and the methodologies that I present can be applied to many marine mammal species, including the local species that many of you study. So many species of marine mammals move long distances across the ocean. Oftentimes this movement is between important breeding and foraging areas. And a very well-known local example that we've covered a lot today are gray whales, so I won't really bore you with more of those details. But one of the main takeaways that I really want you to see from this is that these distances between these important sites are many hundreds or thousands of kilometers apart. And because these important sites can be separated by such long distances, marine mammals can be at sea for many months before they reach these points of interest. And this begs the question, how is it that animals are able to travel across these long distances without getting lost? And so to study this, you can start looking at the field of animal navigation. To navigate successfully, there are two main components that animals are thought to have to achieve this navigation. The first is to have some sense of a mental map where the animal has the ability to assess both where it is currently and where its destination is. The second is the ability to integrate external cues to be able to orient itself within the environment and know which direction it needs to go, and also assess whether it's still on the correct path. However, when we think about navigation within the open ocean environment, at least to humans, there seems to be very little cues that an animal might be using. There are no visual landmarks such as islands that can be used to assess position, and sun and celestial cues might be absent in certain seasons or under certain conditions such as storms. In addition, there is constant wind and wave action that causes animals to drift off their intended path if they're not correcting for these changes. Thus, marine mammals that do successfully navigate within this open ocean environment can hold very important clues to understanding marine navigational abilities and provide us some insights into what they're using to navigate. A perfect example of one such species that undertakes these long-distance migrations across the ocean is the southern elephant seal. So southern elephant seals are a circumpolar species that occur around Antarctica. They spend about 80% of their lives at sea, conducting long-distance foraging trips. And these large species are also known as a term called capital breeders. This means that they use the energy stores built up before reproduction to breed. Thus, they have a really strong incentive to maximize the time that they're at sea foraging and minimize the travel time that it takes to get to these foraging sites. Thus, there should be some sort of selection factor for efficient navigation, which we can study to provide novel navigational insights. The data set that I'm using for my master's thesis is pre-existing data of 312 seals that were tagged from 2008 to 2019. This data covers all sexes, ages, and geographic territories of southern elephant seals around the Antarctic. The tag data collected for these studies provides geolocation data for these individuals across their foraging trips. And so my main research approach when taking this data is to use the travel paths from southern elephant seal geolocation data to provide insights into cues that seals might be using for successful navigation. And to do this, I have three main objectives that I will explore. The first approach is to quantify the consistency of travel paths taken, both between and within individuals. My second approach is to examine the timing and location of directional changes within these foraging travel paths. And the third is to measure the directness of return trips in relation to outgoing trips. The figure that I have here on the right is a visual representation of a subset of the data that I have. So each of the individuals are a different color represented here on the map, and it shows the different tracks that are taken from the Rookery on Kerguelen Islands to some of the foraging areas that they're using around Antarctica. And using some of the objectives here, the findings from that can be used to discuss potential navigational mechanisms that these seals are using and what this might mean for their overall navigational ability. And so some of the importance of these results, I hope to advance understanding into the field of animal navigation, especially animal navigation within marine systems. Much animal navigation work has already been done in terrestrial systems, but marine systems are still kind of lagging in that regard. So hopefully that this can kind of provide more insights. And I also hope that this can lead to conceptual breakthroughs in navigational understanding that can be applied to other marine vertebrate taxa. Many species are navigating, and there are probably very similar cues and mechanisms that they're using. I also want to show the usefulness of analyzing pre-existing tag data to conduct these studies. From a financial standpoint, being able to take data that has already been collected and applying it to new uses can definitely be very useful. And it can also be helpful to study cryptid species or species that cannot be studied in a traditional laboratory setting, which is most marine mammal species. I want to thank all the researchers and collaborators that have worked with me on this project and all of you for listening today. Thank you.
Thank you. Do you have any questions? Okay, then we switch over mics. And Kathleen, any insights into whether you think seals use a different system than whales? Or do you suspect in the end they're all using similar cues in the ocean?
That's a good question. I think that there's probably, in both whales and pinnipeds, they probably are using some sort of navigational mechanism when they're at far distances from points of interest, and then once they get a little bit closer to those areas, they're using a new set of kind of techniques for those smaller distances. I think it'd be very interesting to kind of compare between whales and pinnipeds, but I think that there's very similar cues that they might be using.
Okay. Thanks so much.
Can I ask a follow-up?
Sure.
So what about the role of conspecifics? At some distances would whales, for instance, or pinnipeds queue up on voluntary or involuntary information from conspecifics?
Yeah, so the question was, would they be using con-specific information, so traveling in groups and using information from others? I think that definitely is a factor. For my study species, specifically southern elephant seals, they tend to navigate alone, which is why they are kind of a good species to understand this, because they don't have the ability to kind of follow the group. But obviously many whale species especially are navigating in groups, so that would be something interesting to look at.
Thanks so much. Thank you. And next up, we have Zach Warham.
Hello everyone. My presentation today is going to be an introduction to my work in progress master's thesis. It's titled Using Tracking Data from Antarctic Fur Seals to Identify Patterns in Their Open Ocean Movements. In short, navigating is the act of traversing the environment around you. My goal is to look at how pinnipeds navigate from a data-driven perspective. If we look at our top photo here, we see a picture taken from a caribou camera collar. Surrounding this caribou, we can see a riverbed through 2 wooded forests, hills in the background, and a herd leading the way. The bottom photo is taken from a humpback whale, swimming in the photic zone. But what can you see? Well, other than what could have been the ocean floor, if I hadn't told you it was a whale, we see nothing. So a cue is some form of indicator in the environment which provides information to the animal, such as the position of the sun, and the sensory modality is the sense in the body which responds to the stimuli, such as a change in heat. So if a terrestrial animal has all this information like our caribou, what cues do pinnipeds and other aquatic species have in the ocean? To answer this question, the species my thesis will focus on is Arctocephalus gazella, or the Antarctic fur seal. In general, little is known about pinniped navigation, and there are some sizable data sets available for some species, but the studies using this data primarily been focused on an ecological perspective, not a navigational one. Given a population size being estimated in the millions, the Antarctic fur seal is one of the few pinniped species that has no shortage of data. It has been shown that they migrate long distances between isolated island locations and repeatedly undertake extensive foraging trips lasting several days, which makes them a model species from a navigational perspective. One study showed that upon returning to land, there is evidence of high site fidelity, with about 50% of males returning to within 1 metre of where they left. To be able to have such a positional accuracy in their journeys requires at least one sensory modality that they use to navigate at sea. Here's an illustration of the data for this species that I now have access to. With a little over 1,000 individuals from 2 data sets, with just the Australian Antarctic data centre being visualised on the right here, there's plenty of margin for targeted analysis whilst maintaining statistical power. There's also a significant number of individuals across islands, with the top 6 islands having at least 30 individuals and the top 3 having at least 90. This allows for a strong statistical power between islands on top of that. Unfortunately though, a lot of this data is not very well filtered, as you can see by some random points going across continents there. It does require some work to clean it up, which may in turn reduce the power as these sample sizes slightly reduce. Since this data is gathered from multiple groups over many years, roughly about 20 years for this data, there are various inconsistencies in this data. Through the usage of tools created in R, such as the one to the right here, I will process the data and ensure a consistent analytical approach. Tracks need to be separated into individual journeys, whereby a track is all data points taken during the lifespan of the tag, and a journey, on the other hand, is the path taken after leaving until returning to land again. Unfortunately, there is no defined point in the data that shows when the seal returns to land, so I have to manually inspect each track on a map and determine when it is on shore and when it is not. For example, one track may span many journeys or may be terminated partway through the track. As my analysis is looking at complete journeys and not total tracks, I can use this tool to go through and manually inspect each track in an efficient manner to pull out the journeys and label them as such. Now let's look at my plans for analysis. There's one area I would appreciate any feedback on if you'd like to reach out after today's proceedings. So to briefly summarise each approach, I define linearity as the consistency of direction in each trip. The goal here is to calculate a quantitative value for both the outgoing and incoming portion of their journey, and it is hoped that this will help to indicate details such as the point-based navigation. That is, if they just swim directly for a location or more wander around as they navigate. Total directionality is similar to linearity, except this will be quantified across a grid and consider all individuals. I want to determine how many individuals pass through each grid point and their average, or rather point or region, and their average direction. This will help to map their routes to determine if there are any seal highways, so to speak, and that they all take, and if there are any interchanges that they like to exit on. This could then be correlated back to cues that determine how they know when to take these exits. Nearshore orientation is more a focused version of directionality that looks specifically at the rookery level as opposed to the population level. By determining patterns, such as if they fan out as they leave, or if they follow a specific route, I'll be able to compare between rookeries to determine if the cues used are something that can be used species-wide, or if there's a learned behaviour specific to each rookery. Whilst little is known about pinnipeds in terms of navigation, the Antarctic fur seal has rich data sets, and it's hoped that through this new data-driven approach, these analyses will identify patterns to allow an insight into how they navigate. If patterns can be identified further, research could focus on these specific patterns to try and determine what cues are being used at each point of their journey. And finally, by using the Antarctic fur seal to home in on what works and what doesn't in terms of understanding the statistics of their navigation, we can then focus future research into other species with less available data by using methods that have been defined here. I'd like to thank everyone for your time, and I look forward to discussing this in the future with you all.
Any questions? We'll get you instrumented up, Benya. Go ahead, Josh.
Are you looking at any sort of parameter of decision-making with movements?
Not in this study particularly. Oh, sorry. So the question was, am I going to look at predation in terms of the study? And so now I won't be considering predation. It's just mostly just going to be what's the tracks. And most of these tracks don't have information labelled like that, so I'd have to determine that myself, and I just don't have the time for that given the time frame.
Yeah, I was just curious if you could use the great white shark or Australian fur seal movements, if that data available.
So to just repeat, there was if to use other species such as great white sharks or other species of seals, that would be possible. It's just not something that I've scoped for this project. Thank you.
Thanks, Zach. Okay. Benya Nowak is going to give her talk, and then we're going to take a break. So
Mine's a
just
little longer than 5 minutes, but I was told it was allowed, but I will speak very quickly.
Yes, you're now at 6 minutes, and the timer is started now.
I'm a tight 7, so we'll see. Okay, so hi everyone. My name is Benya Novak, and I'm a PhD student with the Marine Mammal Research Unit here at UBC. And in collaboration with the Norwegian Polar Institute, I'll be studying the movement patterns of Spitsbergen bowhead whales off of Svalbard. But because I'm new, today I'll be presenting some of the work that I did from my past life, studying the movement patterns of gray seals and how they're structured, when they develop, and how you can't teach an old seal new tricks. So I'd like to start off with a bit of background information about both the ecosystem and the animal, since we're going all the way over to the East Coast. So Sable Island is a crescent-shaped sandbar located about 300 kilometers off the coast of Halifax, Nova Scotia. It's about 50 kilometers in length and only 2.5 kilometers wide, which you can see here up on the map. And unfortunately for me, Sable Island is most famous for its population of about 500 wild horses. However, it is also the largest breeding colony of gray seals in the entire world, with the population most recently estimated at about 380,000 individuals. Yeah, and about 80,000 pups born each year. Yeah. So we're doing a bit of photos here. So looking at this male gray seal, you may be thinking, wow, this looks nothing like a harbor seal. And you're right, it doesn't. Gray seals are a lot more like the elephant seals of the North Atlantic, and like elephant seals, they're sexually size dimorphic, where males are larger than females. And in the case of the gray seal, this male here is about 350 kilograms, so getting close to 800 pounds. In comparison, females, on the other hand, are about 2/3 their size, up to 250 kilograms. And for reference, harbor seals are a little bit smaller, only 1/3 the size of an adult male gray seal at about 110 kilograms. So, gray seals also adopt a different strategy for their pupping and breeding season, where both sexes come ashore for an entire month, and they fast during this period. So unlike harbor seals, gray seal pups are also born with their lanugo, or their fluffy and or soggy white coat, to keep them warm while they go from around 13 kilograms to around 50 kilograms, like this not-so-little guy, in only 17 days. So, oh, just wait. Not only do the adults undergo a fasting period, but the pups also undergo a fasting period. Once they're done nursing, they stay on shore for like a week, maybe 2. It's kind of variable. And they also need energy reserves for when they're trying to learn how to catch fish. And so what this means is that their moms also have to have a lot of energy reserves in order to give them to them. And so they come ashore looking a little something like this. Yeah, so you might be asking, what is she eating? So, gray seals are actually considered to be prey generalists, and the males have a more taxonomically diverse diet, whereas females specialize on high-energy prey species. And so, that makes a lot of sense, actually, because the stakes for a female not getting enough energy are actually much, much higher. Gray seals are also benthic foragers, and their diets show seasonal and interannual variation. So, we know what they eat, but where are they eating? From previous research, we know that they're widespread across this continental shelf region, and while their movements are largely concentrated over on the eastern shallow banks that you can see there, it's a little more complicated than that. This region is really bathymetrically complex, and with these currents, and as a result of the bathymetry, it's also really hydrographically complex, and it has some of the highest interannual variability in oceanographic conditions in the North Atlantic. So, in order to figure out how this complexity influences their movements, gray seals were instrumented with tags, like a few other people I heard talk about, that collect highly accurate location data in addition to temperature, light level, and depth data during dives. Those tags, because gray seals have super high sight fidelity, could actually be retrieved. That data could be downloaded and used to calculate oceanographic properties like chlorophyll A concentration, mixed layer depth, and upper water column temperature.
So this has the added benefit that because those data are collected at scales that are relevant to the scales that gray seals are making foraging decisions, we can actually use them to better understand what they're doing. So that's exactly what we did. Here we have 4 gray seals that were instrumented in 2014. The green dots are where they are foraging, and the different colored dots are the different tracks from different animals. And what we did find was that the fine-scale oceanographic conditions are influencing where they're foraging. So what about those pups like Hedgie the hedgehog here, who have to go into the ocean for the first time, which is clearly scary to both of us. I'll give you one more shot with him, because, oh no. Okay, he's back. Well, there's a lot to consider when we think about how gray seals are developing these foraging patterns. First of all, pups have no experience. They don't know where to find fish, and they also don't know how to catch fish. The other problem is that Hedgie can't dive very deep because he had a really good mom, and he's pretty fat, and he floats pretty well. He also can't really hold his breath for that long, which is not that good for a benthic predator. Another thing they have to deal with is that they're slower and a little bit smaller than adults, and there are predators like great white sharks that live really close by and are waiting for them. Well, turns out, despite all these limitations, gray seal pups actually develop these foraging patterns really quickly over the first few months of their lives. And how quickly they develop these patterns depends a lot on a number of things, one of which is how big they are when they're weaned, which depends obviously on how good of a mom they had. And while this is interesting, the problem, like I mentioned before, is that the place they live is really variable from year to year. Well, turns out they don't really care, even if maybe they should. So here what we have is the area where a seal was tagged in 2014 during the winter, sorry, not 2014, 2004. And here's where it went in 2018 during the winter. So 14 years later, we see it doing the exact same thing as itself. And what I have is 20 other examples of animals seasonally doing the same thing as themselves, but they actually do slightly different things than each other. And so what this means altogether is that at an individual level, gray seals may be slow to adapt to environmental change, which is not ideal. So bringing it back to you guys, like gray seals, the distributions of many large marine predators are influenced by the oceanographic conditions they encounter. These results could potentially be applied to harbor seals, like the Atlantic ones you see here, or the Pacific ones. The problem is they're way, way harder to study. They tend to just flush into the water if you try and catch them. They don't come ashore for an entire month, and they also don't return to the same place every single year. Anyways, all that to say, thank you to everyone listening about a non-West Coast species. In addition to my co-authors and funding agencies, I'd like to thank everyone who helped out in the field. There's a very short list of people, and just the ones who had to put up with me. So, thank you.
Do you have any questions? Question in front?
37 something something? Yep.
How did you find it again?
They're just asking how we found that same seal again, and so there's a larger branding program as part of Sable Island, so they have names stamped in their butts, so you know who's who, and it was not necessarily on purpose. This was kind of a byproduct of me doing that other analysis. I was like, hey, it's them again, and so I went through the catalogs back to actually 1995 and found 20-ish seals between 1995 and 2018 who had been tagged, and there's some animals actually who have been tagged 3 times during the same season, and it's the exact same thing, like 19 years apart. They're doing, yeah.
Given that they're generalists so much, it's especially interesting that they show such little variation within individuals in their behavior.
Yeah, so that kind of lends itself to the, oh, sorry. Given that they're generalists, basically why are they going to the same place all the time? And it's kind of, this ties into the fact that we're seeing that interannual variation in their diets. It's actually been seen, and it's kind of more of like an up-and-coming area, that this has been shown in a few different marine mammal species and other large marine predators, that they're using it as sort of like an insurance policy to be like, it's relatively better here than the other places I could go. Okay.
Thanks so much, Pena. We are going to take a break right now. It'll be till 10 after the hour, so please stretch, get something to drink, be back again, and Simon, you'll be first up.
Yeah, of course.
Okay, and welcome back, everybody. We're in the home stretch. Our first speaker back is Simon Surma.
All right, so my name is Simon Surma, as Andrew already said, and I'm a postdoc here at the Institute for the Oceans and Fisheries right here in this building. And today I'll be discussing prey consumption and nutrient recycling estimates for marine mammals in the pelagic North Pacific. So the research questions I'm addressing here basically are twofold. So first off, I was trying to estimate what are the roles of marine mammals in these subarctic ecosystems in the Pacific? Number one, as consumers, particularly of species such as various fish, squid, and zooplankton. So what is the potential for top-down effects due to this kind of predation? And number two, how important are marine mammals as recyclers of nutrients in the water column, particularly limiting nutrients such as iron, given that the pelagic North Pacific is this high nutrient, low chlorophyll ecosystem where you have an abundance of nutrients such as nitrates, but not much primary productivity. So that's in line with the so-called whale pump hypothesis, which I'll come back to later on. So the study area, I'll just give you a quick overview. It's divided in two. So on the left here, we have the Western Subarctic Gyre, and on the right, we have the Alaska Gyre. These are these two current systems in the pelagic North Pacific that stretch from the subarctic subtropical transition zone in the south up towards Alaska, the Aleutian chain, and then the Kamchatka and the Kurils in Russia. So this doesn't include the marginal seas like the Bering Sea, but it includes all of the subarctic waters of the deep Pacific Ocean. And for each of these areas, I have an ecosystem model prepared that will help me answer these questions. It includes estimates of the biomasses and consumption rates per biomass for all the main marine mammal species in this area, and also information on their diet compositions, etc. So these two icons there show the EcoPath with EcoSym, which is the tool that I've used for these ecosystem models. So first off, based on these estimates of biomass and diet composition and consumption per biomass, etc., here we have some estimates of the absolute values of the prey consumption in tons per square kilometer for each of the major marine mammal species found in these subarctic Pacific ecosystems. So the blue bars show the Western Subarctic Gyre, and the orange ones show the Eastern or Alaska Gyre. So we can see from these that the great whales, as might be expected, show up as some of the major consumers up there. So for example, you have fin whales as major consumers of some zooplankton and forage fish species. You have sperm whales playing a major role as the consumers for large squid in particular, but also some fish as well. And you have a number of lesser players there, such as the sei whale, as well as a number of dolphin and porpoise species. And there is also some consumption in the pelagic zone by two pinniped species, the northern fur seal and the northern elephant seal, although these only spend part of their life cycle in pelagic waters, so their consumption is quite a bit limited. So these are absolute values, but when we look at the proportion of the consumption of these species by marine mammals, when we look at that as a proportion of the total consumption of these prey animals by all predators, then we see the real importance of marine mammals in these ecosystems. And as you can see in this graph, we have for several different prey groups, especially the large squid and many species of forage fish, we can see that marine mammals account for, in some cases, over 50% and regularly over a quarter of the total consumption of these groups by predators.
So for example, with these large squid, the large gonadid squid and the boreal clubhook squid, in particular, we see that in both ecosystems. Some of the forage fish found in the western subarctic gyre, so the Japanese anchovy, Pacific sardine, etc. We can see marine mammals, in particular minke whales, as major consumers of those. And we can also see substantial consumption of the juvenile Pacific salmon out in these gyres by many of these marine mammal species, in particular the dolphins and porpoises. And another question I was asking myself was the importance of marine mammals out there as nutrient recyclers. So by consuming these prey we just discussed and then dispersing nutrients in their excreta out into the water column, marine mammals, in particular the great whales, have been hypothesized to perhaps stimulate primary production by improving the cycling of bioavailable nutrients such as iron. So this graph shows, based on two different estimates, high and low estimates of the iron content of the biomass of various species within these ecosystems, we can see that these marine mammals, in total, when taken all together, all the species of marine mammals can recycle up to tens of grams of iron per square kilometer of ocean per year, which is a fairly substantial amount. If you look at the high estimate of the iron content, this is much lower when we consider the lower end of the estimates for the iron content. But when we look at this relatively, as a proportion of the iron recycled by all the living organisms in these two ecosystems, we can tell that marine mammals actually become quite insignificant players, especially when compared to zooplankton, but also squid and many species of fish. So these other organisms, due to their high turnover rates and higher overall biomass and consumption, recycle a lot more iron than the whales do. This may have been different before, and commercial whaling decimated many of these species of whales in the North Pacific, although many of those are already recovering. But other analyses in the Southern Ocean have suggested that even pre-whaling biomasses of whales would have left the marine mammals as relatively minor players compared to smaller organisms as recyclers of nutrients in the pelagic ecosystems. So just quickly go through my conclusions. So marine mammals, especially cetaceans, appear as notable consumers of fish and squid in the pelagic North Pacific, and less so for zooplankton, which are relied on by many different predators. This suggests the potential for top-down effects at high marine mammal biomasses. However, marine mammals appear to play a relatively minor role in iron recycling compared to zooplankton, fish, and squid. Finally, I'd like to thank all the partners in the International Year of the Salmon project, which forms the umbrella under which I've been doing this research. And I'd also like to thank you all for listening, and I'll be happy to take your questions. Mm-hmm.
You know, we have so many more humpbacks here now than previously, and they eat a lot of forage fish and krill, and our concern has been, you know, are they eating more food than we really have? But we've kind of made ourselves feel better that they're putting nutrients back in. Are you saying that they're not really contributing probably a significant amount of nutrients back in to the ecosystem?
Well, this analysis didn't really cover humpback whales because they're not found as much in the open Pacific compared to the continental shelves and slopes, as we've seen quite abundant humpbacks in many parts of the coastal ecosystem, but further out in the ocean they're not as prevalent. But to go back to your question, I actually looked at that in my PhD for Haida Gwaii, so the Queen Charlotte Islands off northern BC. And it seems that up there, humpback whales consume large amounts of herring, for example, But although they may contribute to some extent, they're certainly not the major driving force behind the declines of herring and other forage fish that have been observed on the coast from the continental United States up to Alaska through BC. But I think there's still a lot more research to be done on the roles of marine mammals as either top-down impactors on their prey, but also potentially as keystone species that may stabilize fluctuations in the ecosystem through their effects as predators. And there's still a lot of research to be done on the whale pump hypothesis, I think. This kind of data certainly casts some doubt upon it, and it's been seized upon very heavily in the media. It's certainly a cool idea, but it seems like right now it needs to be re-examined in more detail globally, not just here in the North Pacific.
Thank you, Mythbuster. Thanks very much, Simon. And our next speaker is Julia Adelsheim.
I know everyone's been dying for a physiology talk, so here it is. But don't worry, it's not too intense. So my name is Julia Adelsheim. I am a master's student here at UBC in the Marine Mammal Research Unit. And I'm going to speak a little bit about my master's research in progress, which is a bioenergetic model for a sea otter. And I'll explain what that is, don't worry. So for just a bit of physiological context, something that's unique about sea otters that is different from their marine mammal cousins is that they don't have a blubber layer. Instead, they have very thick, dense fur that traps air and is warmed by their body. I think we all know this. But because of that, like the lack of built-in storage, they have really high metabolic rates, and that in turn means that they have really high food requirements. And because of that, they have a disproportionate impact on their local environment compared to other species within that immediate environment. And they can especially have a high impact on their prey base, which is basically the definition of a keystone species, which this is that classic example. And so my research is delving a bit deeper into what happens if a sea otter is specializing in their diet, and answering questions like how much food does a sea otter need, and as they change their diets, how does that affect their overall food requirements? So I am building a bioenergetic model, which is basically a statistic that represents all of the energetic inputs and outputs of an organism that can tell you the food or energy requirements based on conditions set, whether environmental or reproductive, age, sex, what have you. And there is so much energetic and physiological research done on sea otters, but no one has put them together yet for a sea otter. So this is my attempt to do that. There are three main components that I'm looking at. First off is just the energy costs, the cost of growth, the cost of basic metabolism, and then also the losses of digestion and feces and urine. And then in addition, prey quality, basically the quality of the prey item is dependent on relationships between how much energy that prey item will give them, or also the energy needed to handle that prey item. So it's really different diving down and digging up a clam versus picking a snail up off a rock, right? And the quality of that prey is really different. So I'm trying to look at those relationships to figure out the energy requirements and therefore the prey requirements based on diet. So this is just an example of an output. This is just a generic one that I have made, and you can see the differences between a male, a female, with or without a pup, and how they vary based on the age, which is on the x-axis, and then the energy requirements in kilojoules per day on the y-axis. But that is not as interesting. But there's a lot of things you can do with the bioenergetic model, which is why they're really great tools. So for example, other people today have been talking about daily activity budgets. So if we just have an arbitrary example, a 7-year-old female sea otter with a pup, she's spending 60% of her day resting with that pup, 34% of the day foraging for her to then transfer that energy to the pup. And then 6% is doing non-foraging activity like grooming or swimming. But then what happens if we change the conditions and say something happens and the available food in that area for that individual sea otter decreases by 20%? We can run the model with that different conditions and look at how her daily energy budget really changes. And she's spending a lot more time foraging instead of resting, and that has real energetic consequences for that otter. But also, I just want to say why I think you should care about my thesis as much as I do. It really is a useful tool that does not exist for sea otters. They exist for stellar sea lions and walrus and other things like that. It also just helps us quantify sea otter food requirements and sort of look at how a prey item may limit the like a sea otter population expansion, like Linda was talking about earlier on Vancouver Island, or we could look at how sea otters are in turn impacting the prey bases that are there, which then just overall help us inform management and conservation policy. So I think it's great. And with that, I'd just like to say thank you and acknowledge Dave Rosen, my advisor, and my committee, Nicole and Andrew. So thanks so much.
Because they don't have that built-in storage and they're like floating around in cold water, so they're having to constantly replenish their basically energy source. And they lose a lot of heat through the heat increment of feeding, which is the heat we give off during digestion. And that is like a very important energetic factor for sea otters. So yeah, does that answer your question? Okay. Yeah.
Just curious if you have any components of this modeling algorithm that would allow you to look at the impacts of climate change over a longer term, like water temperature and maybe other things that might feed back into.
Yeah, I haven't specifically done any of like the environmental part of that, but I think, that all affects what prey is available, and we saw how tide pools are affected by heat waves and things like that, so it's absolutely relevant, yeah, for sure.
I'm going to cut you off there.
Yeah.
Thank you. This awkward part here of switching over from MC to giving my own talk. So coming up on the screen in a moment, there we go, is the cost of living, which is a concept that I know everybody in this room is very keenly aware of. I think we hear about it every single day. But in terms of marine mammals, most people assume that all marine mammals have a high cost of living. And however, we found in the end that it's not quite true, that marine mammals can be categorized as having 3 different costs of living: low, medium, and high. And I'll show you in a minute what I'm talking about. So this is sort of how I spent a lot of the pandemic, was working from home, building a huge database and working with Kate Colson on the other side of North America, who was also locked down, and worked quite well together to build a database that I think can answer a lot of basic questions about marine mammals, including resolving some of these fundamental disagreements. One of the disagreements has been over whether or not they are like terrestrial mammals, or are they different? And what we explored here are questions about how marine mammals have co-evolved with their prey, and some had to get faster to eat prey that were constantly getting faster themselves, and others went the other route to eat things like seagrass. They didn't take a whole lot of ingenuity to catch and process. Let me give you an example here of 2 species. 2 species of dolphins, a striped dolphin and the common dolphin. And both of them, so here we're looking at the Atlantic Ocean, they're both about 80 kilos in terms of a female. If you look at their diet, there's a huge difference in the average energy density of their diet. The striped dolphin, about 4.5 kilojoules per gram, and compared to 6.8 for the common dolphin. is 33% higher. So why are they eating a higher energy diet? Well, when we look at their muscle performance, they've got greater mitochondrial density and higher lipid content. Essentially, they're like racing cars. They need high octane fuel, and they can't get by eating low energy prey. So we categorize them as being high, and here having a medium cost of living. Where this comes into play is going back to some early work that Stu Innes and Dave Levine and others did, and Kit Kovacs as well. And on here, we've always been looking for these common relationships, assuming that all marine mammals should fit single straight lines. They're just different by a function of the body size. So here in this x-axis, I'm plotting the body mass. It's in a log scale against food consumption, the kilograms per day a marine mammal needs. So we found data here for 34 species of marine mammals. And here, when I plot the 34 species, this is what it looks like. You see, yes, it's there's a linear relationship, but it doesn't fit a single line. Here's what Innes et al. Said, and this was their equation. I'll show it with a green line. And I and many others around the world, we use this equation to predict the food requirements of marine mammals around the world. But you can see that it clearly is not a very good fit to the data. So we dug into this further, going back to the cost of living concept, and we're able to categorize these 34 species, and this is what it looks like. If I fit to the species that are low, that's in the yellow, the top line, the species that are categorized as medium, they're at the bottom, and you might be wondering, where do the high cost of living species fit? They turn out to be a little bit here in the middle, grouping closer with the lows. If I look at Stu Innes' equation, this is where it fits. And the most incredible thing for me is then going through their paper and looking at all their species, they use low cost of living species to fit their model. If you look at the work from Terry Williams and others that have been arguing, no, they've got much higher cost of living, higher energy requirements, they've been using high cost of living species, California sea lions, killer whales, and others. And so it turns out that the basis for this argument has been we're using different groups of species. But when you put them all together, there is no single line that works. So a quick explanation, well, those that are high, they're built for speed. They're built to swim fast, and they're chasing after prey that are also built to swim fast, and often they've got muscles that are fueled by lipid. On the other hand, the low ones, I look at them being the stealth, they eat slow, or prey that barely move at all. They tend to be low lipid prey. And then the mediums are what I call the average speed and the average stealth, so the generalists. They're the ones that are more like terrestrial mammals. So these two have to eat more, and this group can eat less. And so in the end, it comes down to trying to figure out, so what's the cost of living of all these species, many of which we've never studied? And so we've come up with a system to categorize them, and I don't have time today to go through it, but it's a fairly simple system to use based on some basic characteristics that can be applied and scored. And when we do this, we find that there are cutoff points, and so we have a scaling that goes from the bottom up to a high of 15. And you might be curious to know, like, where does your favorite species of marine mammal sit in here? Are they all high cost of living? And on here, we've got at the bottom, elephant seals, gray whales, Australian sea lion, the dugongs, the manatees, the walrus, and look at that, the sea otter. And I realize some of you are going to say, no, but didn't Linda say they just eat things that don't move very much? They waste energy, but they get by eating things and digging things out. At the high end, yes, there's the killer whale. We've got Dall's porpoise, sea lions, fur seals. And then in the medium, the ones that are more like, and both these groups have to eat a lot more as a result, and those that are eating less we've got the humpbacks, belugas, ring seals, harbor seals. And so you can see in here that things do group out, and it's a different way to think about marine mammals than the way we've been treating them. They don't all fit a single line. So marine mammals cluster into 3 types of lifestyles corresponding to high, medium, and high cost of living. They're independent of body mass and phylogeny. And these other two, they have to consume so much more compared to the medium. And what we speculate is that those that are high and low may be less able to adapt to environmental change. So we're in the process of writing this up, and I've got a sabbatical coming up, and this is going to be a top priority to get this work out. So there's the question for you. Is your favorite marine mammal a high maintenance? Anyway, thanks very much. So I'm happy to answer any questions as Marty comes down to get mic'd up. Am I muted? Oh, Dave.
When you're talking about cost of living, are you talking about calories or mass of food?
In the end, it is basically a relationship between the caloric value of the food. That is one of the indicators, but it's also the energy that animals are putting in to acquire it. So in terms of scaling it out, we've come up with a simple index that we add up the costs and the benefits. And certain energy density is one of the factors, but not the only one. Oh, I will repeat the question next time. Gosh, it's been a long day. I think we're ready for Marty, Helena. All set? You guys are good?
Thanks. And where do I switch here?
You're good.
I mean, where do I switch slides? Here? Or do I have a remote?
Yeah, no slides. You don't have slides here. Sorry, 1 second.
Yeah, but where do I switch them? Here, or do I have? Okay, thanks.
You can use the remote, sure.
Oh, okay, so there is a remote.
We're not gonna
You're just trying to trick me.
Well, yeah, we're okay.
Okay.
There you go.
How do I use this now? Are we ready? No. Oh, you've gone through my there it is. Start at the first slide.
We need you back for the camera.
Oh, I can't move around? How am I going to point things out? Online audience. If you cared, you'd be here. That's right. All right, you guys. I'm going to have 5 minutes, and I have a lot to talk about. You may or may not know we've gone through a few changes, but we're still here. That's the Vancouver Aquarium. We are a slightly different format, but all those lovable characters you've known to grown up to love and tolerate are still here. And we even work with Rosen still, which is kind of nice. But we try not to. Just summarizing a couple of things we've done over COVID here. I'd want you to concentrate on this and take a good long look. No, we did a bunch of research, right? Which is great. We kept things moving along. We've been working on a number of different things. Zach's got a project going with us. Our big in-house project that the veterinary department is involved in is with Rhea Storland, who's one of Andrew and Dave's PhD students, and who also completed a master's with us. And she's really working on how hearts work in diving animals, so specifically our stellar sea lions for her PhD. She worked and did a master's on basic cardiac function in our animals, which was great. Now she's gone on to do a PhD, or doing a PhD. And she's one of the kind of interesting tools that she's helped us develop is the use of these implanted cardiac monitors. So this is a procedure sped up a little bit. That's me drinking my coffee. That's Rosen not doing anything. And that's this is how you implant a little cardiac monitor. So that was a sterile prep. There's Rosen not doing anything and getting in my way. And that's it. And if you notice, the animal actually was asked to come in and put that cone on her face on her own. So that's completely an unrestrained procedure where she knocks herself out with the gas and goes along and eats after you, and away we go. Once the implant's in there, Rhea can use that. It looks like a little USB stick. That's about the size of this thing. It's on the X-ray there. I can't point to, but there it is. Hold on, taking a deep breath. So that instrument can record heart rate, ECG for about 20 minutes or so. Rhea can go up to that animal and from a distance of a few feet can download that data and use that to analyze for a variety of different functions. Personally, we're very interested on how cardiac function is changing in our sea lions, because they kind of, unfortunately, we've got some old animals now, and heart disease is a big issue in aging animals, and sea lions included. So we like to monitor them. Ria likes to do cool sciencey stuff. On the topic of aging animals, we've got kind of another, interesting case, and this is Hermes, who's actually an ex-UBC harbor seal that used to live here many, many years ago, and now has been sort of retired at the Aquarium. But he's developed kidney stones, which is unfortunately a relatively common thing for seals and sea lions to get. They get that in the wild. They get it in human care as well, especially as they age. And so now the problem with kidney stones in these guys is they have a very, very complex kidney. It's sort of this multi-reniculate kidney, lots of little channels and passages. And the traditional ways that we get rid of kidney stones in humans and pet species, especially using kind of an endoscopic technique, don't really work. But we've collaborated quite a bit with UBC in a number of different ways, including through the medical department here. And we were turned on to a rather cool new technology. So this is our seal Hermes getting a CT to kind of show you his kidneys in that, and that's his one kidney. You can kind of see the elaborate network of stones that have developed there, and how tricky it might be to get into those things. So this is your comparative medicine building here. I forgot what it's called, CCMWCC, MMC, I don't know. But it's got it has a kind of a cool setup for a fluoroscope. And then the really cool part about this is we were turned on to this machine that's made by a company called Sonomotion, which is called breakwave technology. And what it does is it uses very precisely guided high energy sound waves. So this is an ultrasound image, so it's guided by ultrasound. And you can see the kidney stone being zapped and kind of disappearing in front of your eyes here. Now, it does take a relatively long period of time, so Hermes is going to have to undergo a few different procedures, but this is a very new kind of sophisticated technique. It's only been done in about 50 humans worldwide, so it's still not fully FDA approved, for example, so it's still in clinical trials in the human field, but because of our collaborative nature with UBC primarily, we've been able to kind of take advantage of these kinds of techniques and learn to develop what we do and keep getting better at what we do, even during COVID, which is fun. We've been out in the field a few times, working primarily with our DFO partners and Paul specifically and his team. Of course, we do a number of different things. I guess one of the coolest things we're involved in is this gray whale named Chocolate, who had a satellite link transmitter attached on the American side, and it got infected. You'll see there's going to be the whale's going to come up here in the video here, and you can see the tag kind of causing this saddleback or saddle patch lesion on its peduncle. You can see that's the tag there, but the inflammation associated with that is actually coming out on the other side. So there's quite a degree of infection. You can kind of see it there, and you can kind of see the infection on the other side. So based on some work we did with J50, developing a technique to deliver antibiotics to large animals in the field, we did go ahead with Paul and the team and delivered some antibiotics. And this is how you treat a gray whale on the coast. Now, unfortunately, we were limited a little bit. You can kind of see the dart. There it is. You see? Now, Chocolate did feel that, obviously. And we also got a poop sample at the same time. So you're welcome, all you poop guys. And now the only problem is this is a big whale with a relatively small gun, so we had to get 4 darts, 4 10-milliliter darts into this animal over the course of 2 days. So the satellite tracking device that was implanted did help with that because we could find Chocolate, but that's kind of the way that went. And of course, our disentanglement program for pinnipeds, also in collaboration with DFO, has been ongoing. And this is from March of this past year. This is at Deltaport on one of the mooring buoys. This is an adult female stellar sea lion that's got quite an entanglement. We're launching the dart from the DFO boat. You can kind of see it there. She had the nice kind of behavior of staying on that, which is nice, on the buoy. We do work with the animals if they do. Oh, the video stopped for some reason, but anyway, kind of get the idea, I suppose. Remember that time we checked the video and it worked? But Oh, maybe that might just be it. Anyway, we'll show it again. But essentially, we do work with the animals if they go in the water as well. Of course, if they stay on something dry, it helps a lot. So essentially, you go in there and deal with that animal. And in this case, it was monofilament fishing line, this animal. We do get a lot of packing straps, but this was quite a bad entanglement. The plastic had gone quite deep into the animal's neck, had started to grow over. So we had to do a few different incisions and pull that out, and away we go. But that's it for me. Just a quick update from the aquarium. We're here.
Thank you, Marty. Any questions? Do you get any sense, Marty, of increased incidence of entanglement, or is it just a constant background noise?
So I don't have to, do I have to repeat that question?
You will have to.
Oh,
No, you don't have to repeat it.
because he repeated it. Okay. Okay. I'm starting to get this now. Okay, so no, we are not getting ideas. Long story short, yeah, I think it's constant. Oh, now I go over there. This is so confusing. So yeah, no, it is normal kind of background. In some respects, we think we see less, and I think unfortunately that was mostly because people weren't on the water for the last couple of years looking for things, because we were always relying on folks out there. And Brianna actually just mentioned that they've been keeping tabs on a few entangled pinnipeds. So that's good information to pass on to us, and we'll work with Paul and get to those animals. So I think it's unfortunately kind of a steady long-term thing. Yeah.
Thanks so much.
Okay. Don't you do anything with that? Do I have to turn it on or anything?
And next up, we have Cassandra Girdlestone.
Thanks, Andrew. Hi, everybody. My name is Cassandra Girdlestone, and I'm the research assistant at the Marine Mammal Rescue Center. Just to tell you a little bit about MMR, it's been in operation since the early 1960s, and it's the only facility of its kind in Canada, and we focus on rescue, rehabilitation, and release of marine mammals. So just over the past few years, we've had a little bit of an identity crisis going on. We started out as part of the Vancouver Aquarium. Then we became part of OceanWise. Then we became Vancouver Aquarium, an OceanWise initiative. And now we're back to Vancouver Aquarium. But through it all, we've remained the Marine Mammal Rescue Center. So today I'm just going to give you a little quick summary of what we've done over the past year at MMR. Our volunteers, we had 166 volunteers working over 18,000 hours to get us through the season, and we absolutely could not have done what we do without them, so we're grateful. As all of you here have experienced some changes over the past couple of years, so did MMR. The one big thing we noticed quickly was a lot of differences in the calls coming in. So reporting animals, as people moved outside where they hadn't necessarily spent a lot of time, they were experiencing new things. So we had a lot of public education, explaining what's normal, what's not normal, and telling people about the natural history of harbor seals and other animals. So that was interesting. We also had a smaller team, so we didn't have any recruitment of new staff or volunteers during that time, which led us to greatly appreciating it when they were able to come back. So once again, we brought on some more volunteers this summer, and practicum students have returned. So any of you going to UBC, the APBI program has a practicum placement at MMR, so check that out if you want to join us next season. As far as animals reported, we've had about 366 cases reported and counting. We're still having calls come in. And over 266 of those were harbor seals. I've got a list here of active responses, and that does not mean that those animals were rescued and admitted to MMR. It just means that we made a plan and in many cases tried to execute that plan, but nature doesn't always cooperate, so the animals don't necessarily stay where they were sighted. So we will continue to try helping them, but we're not always successful. And as Marty mentioned in his presentation, one of the disentanglement trips, we've got a few more listed here that we carried out over the past year in conjunction with DFO. And these are just some facts
facts about our pups that we had in this year. So the one I'd like to draw your attention to is Lanugo pups. We had 19 of those come in this season, and Lanugo pups have white fluffy fur. They're really cute, that thick fur that they usually lose in utero. When they're born with that, if they have about 50% or more coverage, that's typically considered to be premature for harbor seal pups. So an 84% survival rate is pretty good for that. And then lowest admit weight was 5.4 kilograms. And just to give you a point of reference, the average birth weight is typically somewhere around 9 kilograms. And the cutest seal, well, all of them. How could you ever pick just one? And that being said, although I don't have nearly enough cute seal photos in here, you can go to mmrpatients.org to get your fill there. So, and this is a map. All the red dots are locations where seals were rescued from this season. And if you look at that yellow circle up to the right, that's Harrison Lake, which is a freshwater lake. We always have a couple of cases or so up there every year, and would definitely like to look into that a little bit more and see what's going on. So if anyone's interested in doing a little research to learn more about that population, that would be great. So this year we had a few projects on the go. We've definitely increased capacity for oil spill response. And as I mentioned, we've had increased volunteer recruitment and training going on. We've done our disentanglements, and we have a few research projects which I'm going to give you the highlights of coming up next. So the first project is the antimicrobial study done by Daisy Hahn at Tufts University. And she is using fecal samples to identify specific virulence genes that are encoding for resistance. And she's comparing those with the results of culture and antimicrobial sensitivity testing. And she's using rectal swabs to get that information and doing swabs at 3 points in time over the course of the season. The next study is corneal ulcer microbiome swab study done by Zach Warham, who you heard from earlier. And he is trying to identify a relationship between eye injuries and a change in the microbiome associated with those injuries. And he's doing so by taking eye swabs from the harbor seal pups. And the last project we've been working on is avian influenza sampling. So because that was detected earlier in the year and continues to be present in BC, in conjunction with BC Ministry of Agriculture Animal Health Centre, we're sampling all the seal pups that have passed through MMR this season. And that involves 1 nasal swab and 1 rectal swab per seal. And those results will be analyzed at a later date. And then in conclusion, just want to thank Department of Fisheries and Oceans for providing funding so we can do what we do, and as well as all of the organizations and people who help us and support us in our daily work. And thank you for listening.
And thank you. Do we have any questions? Question over here.
Just out of curiosity, is there a theme for the pup names this year?
And
so the question was, oh, I'm coming, sorry. The question, hey, don't lump me in with that guy. So the question was, is there going to be a naming theme for this season's the seal pups this year? We haven't decided what it will be coming up. The 2022 season, we had sweet treats. Sorry, I just drew a complete blank for a second. Sweet treats. So we had like cream puff and banana split and Nutter Butter, things like that. And we also work in conjunction with IWNCC over on Salt Spring Island, and they typically have a separate naming theme. Theirs was Seaweeds of the World this year. So.
And if someone wants to volunteer, what's the process?
Definitely go to the Vancouver Aquarium website and look under MMR and volunteering opportunities, and just send an email to the volunteer coordinator. And there's a bit of a training process and everything like that, but yeah, we'd be happy to have more help.
And does it help to say you attended the BC Marine Mammal Symposium?
I can't
can't hurt, right?
Okay, thanks so much. And next up, we have Dana Price. All set, Dana?
I believe so.
Thanks.
Okay, hi everyone. I'm Dana. I'm a first-year master's student here at UBC in MMRU and also the Ocean Pollution Research Unit. My thesis is on contamination in BC's sea otters, and today I'm going to talk a little bit about microplastics in the sea otters. First, I just want to thank all the people who are making this research possible, my supervisors, Andrew Treitz and Juan Jose Olava, and then also connections at the DFO, Stephen Roberty and Paul Cottrell, who helped provide the samples. So, I'm assuming you've all heard a little bit about microplastics, but I'm going to do a little bit of background just in case. They are quite a hot topic in the news these days. The general definition for the microplastics is that they're under 5 millimeters in length. They come from all sorts of different sources. They can come from synthetic clothing. They can be primary from the plastic production process, little spheres called nurdles, which is a fun word. Also quite often found in water samples is little fragments from tires, sort of as you drive on the road, little bits of the tire shred, and so that also runs off into the aquatic ecosystem. And microplastics are just about everywhere. They're in the air, they're in the water, they're in the sediment, and they've even been found in human blood. So that's fun. We're still kind of figuring out, it's not super known, the connection between microplastics and specific pathologies, although studies have found them to absorb harmful contaminants and also bacteria, so it's definitely worth studying how much is in animals so that we have a better sense of how to protect them. So I study it in sea otters. As Julia mentioned before, sea otters eat a ton of food, and including shellfish, which there have been many studies that have found microplastics in shellfish and sea urchins as well, and even fish. So they're eating a lot of food, and we know that their food has been found to contain microplastics, but what we don't know is how much microplastics are in the sea otters. So that's what I'm trying to figure out, if it does indeed transfer. And it's still being determined whether microplastics bioaccumulate or not, so it's good to set a baseline for the amount that's in the sea otter gastrointestinal tracts. So I don't have any fun results yet. I'm still at the early part of my research, so I'm just going to walk you guys through sort of what I'm doing at the moment. So I start off with sea otter gastrointestinal tracts that I've gotten from Stephen and Paul at the DFO, and I thaw them. And by the way, these are all from sea otters that have been found dead. This is in the necropsy process that the samples were collected. No live sea otters involved. So I separate the organs, so I separate out the stomach and the intestines, put them in different beakers so that I can start liquefying them. I use potassium hydroxide or sometimes hydrogen peroxide if they're a bit trickier, and sort of melt down the organs. So in the photo here, that's a mostly digested set of intestines. So that way it's liquid and I can run it through a very fine filter. I use a 10 micron polycarbonate filter to sort of catch any particles that are in those tissues or in the contents of the organs, and hopefully catching any microplastics that would be present. And then looking at those under a microscope, I will measure the length of them, note the color, any sort of defining characteristics of them. This one is not from sea otters, it's from a shorebird while I was calibrating the microscope and program on my computer, but that's what it might look like when I find anything in these sea otters. From there, the next step is to do chemical identification. We want to confirm that any of these particles are indeed plastic, and if so, what type of plastic they are. And that's done either through FTIR spectroscopy or Raman spectroscopy. So currently, I'm in the dissection, digestion, filtration stage, sort of going on simultaneously, and hopefully in a year or two, I'll have some fun results to share with you all. But that's it for now. Thank you to everybody who's been involved in this research and made it possible.
Any questions? Question at the back, and you get to repeat the question.
So the question is, did I find anything weird in the stomachs or the gastrointestinal tract? And the answer is yes. So in one of my samples, I just found a slightly ridiculous quantity of worms embedded in the intestines. Caught me by surprise, but a lot of small worms that were just embedded in there. And I've also found some undigested crab parts too, but that's about it. Yes.
More a comment just in research on anti-fouling paints. It turns out the paints all are sort of embedded in a sort of plastic matrix, and there's a number of papers suggesting marine microplastics from paint degradation from vessel hulls is a considerable problem.
Thank you for that. Yeah, that's a great point. So,
can you summarize that point?
Yeah, just for the online people, it was just brought up that anti-fouling paint has plastic component to it and is contributing to the microplastic issue. That's a great point. Thank you.
Awesome. And you can have my job next year to repeat questions. That's very good. Thank you. Our next speaker was unable to be here, but he sent a recording. You can hear more about microplastics from Juan Jose Alava. Can we just play that?
Good morning, everyone. This is Juan Jose.
Is there no audio?
I am connected virtually today with a pre-record presentation on microplastics in marine mammals inside some exposure and vaccination. This work is possible to the support and assistance of my graduate students, Carly McMullen and Diana Price. So, microplastics, what are they? So we basically live in an age of plastics, and scientists call this the plasticine. Because there is no corner on the global ocean, planet Earth. Planet Earth, our blue planet, are being exceeded. This is one of the greatest environmental problems that we are facing today. We know that these particles are floating in the ocean from 5 to 50 trillion plastic pieces in many areas of the world. The plastic production, for example, in 2020, was like 370 millions, and that's basically equivalent to more than 2.4 or about 2.45 million blue whales, assuming basically a mass of 150 metric tons. And that's of concern. We know that large plastic, microplastic, basically is affecting many top predator charismatic species. And we can see that in terms of the mutilations, the asphyxiation that they are causing, the entanglements, the diaphragm obstructions, and many other kind of impact that is affecting many species, including marine mammals, seabirds, and sea turtle. And also macroplastic or large debris, large plastic are vector of pollutants like persistent organic pollutants, hydrocarbons, and metals, and also biovector of biological pollution like bacteria, pathogen, and invasive species. But microplastic, basically, this is the issue, the insidious pollutant that we see, and they are basically everywhere. And basically, there are plastic particles smaller than 5 millimeters. And they can be categorized as a primary microplastic, which are human-made. They are basically feedstocks, pellets to produce large plastic or macroplastic, and of course secondary microplastic, which are basically the plastic that is breakdown products of large plastic, and they can go from many human activities in the ocean and the land. This really captured, this photo by David Rosen really captured our imagination that basically they are everywhere, and they are in the water that we drink, in the food that we eat, in the seafoods, and in the atmosphere. So if of concern. And the peer-reviewed literature already basically depict the fact that many marine mammal species are exposed to microplastic, and that include dolphin species, big whale species, pinnipeds, baleen whales. So there is the armor races between microplastic and the species that are being contaminated in different parts of the world by microplastic. But how this exposure basically take place in the marine environment. So you understand that basically we need to think about the multiple media compartment, the multimedia environment, right? The atmospheric air, the ocean environment, the water, the sediment, the suspended solid, the effluents from outfalls and runoff coming from agricultural area. So everything come together in the habitat of marine mammal, of salmon, of many species of fish, and that can really permeate food webs, right? And also keep in mind that there are ocean currents that can take plastics or microplastic from one part of the ocean to remote oceanic areas of the world. So this is really the pathway of exposure, right? Thinking about the environmental compartments in the marine environment. And that facilitates, of course, bioaccumulation and potential biomagnification, something that we study here. And also important to keep in mind is the foraging strategies of marine mammal. It's important because we know there are many kind of tactics, like, for example, orcas and pinnipeds, like the iconic killer whales, southern resident killer whales, and stellar lion, they feed on fish and salmon, right? So there's raptorial feeding because they target, they chase, and they capture, and they snap. In the case of killer whales, they swallow, and the stellar lion can rip it apart and also eat the salmon. The baleen whales, of course, we know that are filter feeders. They basically swallow a lot of water with zooplankton and anchovy, and then they push with the tongue the water, but in the baleen, basically they can retain the microplastic or plastic particle that are in the water. And also the prey, the krill or the anchovy can contain microplastic in the bodies. So that's a specific feeding strategy by baleen whales. And then we have the bottom feeders like gray whales, right? That they basically scoop large amount of sediment, a cocktail of contaminants are in the sediment, and that include microplastic and also macroinvertebrates or little critters that are part of the diet. Also, all the pinnipeds like walruses, of course, they can also ingest microplastic through benthic macroinvertebrates, like similar to sea otters, that they feed a lot in microinvertebrates, right? And that can be a exposure pathway for microplastics. So we know that, for example, cetaceans have 4 stomachs, right? So they're polygastric, and that means that the particles can take a little while to cross all these compartments. And that's an important feature to keep in mind, because the anatomy of the cetacean are complex, and that's important for the retention time and the elimination rate. The retention time, for example, is inversely related to the elimination rate that we see here. And this is basically important parameter when we talk about the accumulation of plastic particles, right? And if we think about the fourth stomach where the particle enters, then we have the main stomach or fundic chamber. There is group of connected channel, where basically are the region in which food and plastic can be retained. So it can be some microplastic retention. And then, of course, we have the pyloric compartment or pyloric chamber, right? And of course, potential defecation of plastic particles. So the highest the retention time, the lowest the elimination rate, and the lowest the retention time, the highest elimination rate. So it's basically a function of elimination. And that's basically the parameter that dictate the accumulation in the GI tracts, right? Here at the Institute for the Ocean and Fisheries and the Ocean Pollution Research Unit, basically we predict and model also the accumulation of these particles in the global ocean. And for that, we use databases like Litterbase, which is a database containing the data for peer review, literature, portraying the distribution and concentration of microplastic in the global ocean. And also we made use of the database EcoBase, which is basically ecosystem models that are already available in this data set. And we married basically these two data sets to basically try to predict using EcoPath and EcoSeed modeling with EcoTracer. It's a routine to track bioaccumulation of pollutant. We did that for 16 marine mammal ecosystem, right? In terms of the plastic pollution footprint in the global ocean. Some of the results show for this one is the global footprint of plastics, right? We have high levels in red, lowest in yellowish and white colors. Basically, we found that most of the food webs that we study exhibit a high concentration in top predators, like marine mammals or predatory fish and seabirds. And that happened basically in all the ecosystems that we assessed for this effort. And that's important because when we look this at the specific level, for example, here in the Northeastern Pacific, we see that in the food web of the orcas, in this case, the transient or the killer whales orca, here we have the concentration in the x-axis in microplastics per grams per kilograms. Basically, the top predator exhibit the highest level, and then we have the baleen whales like humpback whale, and of course we have the zooplankton at the bottom as the entry point of microplastic, right? So high levels in top predators, not surprising according to this modeling, but also we can calculate the trophic magnification factor, which is a metric bioaccumulation criteria to see whether or not this might biomagnify in food web. And we do that by relating or correlating the logarithmic concentration of microplastic versus the trophic level. And here we show that the concentration of microplastic increases as the trophic level increases. And that's basically the slope. This number is basically the slope, right? In logarithmic scale. The same happened, for example, for the Alaska and Aleutian Islands, in which we basically, the top predator in the system here are the orcas. We can see, for example, the orcas are not excluded. They are also exhibiting high levels, and that's because basically they are feeding on macroinvertebrates in the bottom. My student Dana Price is studying, is conducting a food web modeling and studying geotracks or other to examine whether or not there are microplastics. She will tell you more, or she already tell you about that. And also, humpback whales are not behind. Again, trophic magnification factor here is the case, right? Increase in concentration as the trophic level increases. In tropical system also, we observe the similar scenarios in terms of the accumulation and dolphins, of course, lower level compared to the northern regions or ecosystem in the northern hemisphere, but still we see this scenario of positive significant relationship as the trophic level increases, right? High concentrations. There are several factors affecting the bioaccumulation, and that's a function, as I mentioned, the foraging strategy, feeding preferences, the amount of phytoplankton available or bioparticles in the ocean, because zooplankton sometimes cannot discriminate between phytoplankton and microplastic, and sometimes they eat more phytoplankton and less microplastics. So there is like a competition between these kind of particles. And also the color, the density, shape, or composition of polymer is an issue. That's part of the physical chemical characteristic for the exposure and bioavailability. And other factors like biofouling, and of course the detecting parameter here is elimination rate, which can be species specific.
Presenting on behalf of the Canadian Wildlife Federation today. I'll be speaking on marine mammal entanglement.
And years on the folks on the water. We work with a lot with First Nations as well. They're in key isolated areas and are a real big part of the response network. One big piece that we've kind of reintroduced after COVID is training, and we really have 4 kind of cornerstones. One of them is marine mammal acoustic deterrent training, which we're realizing is really important with all these recent things that have happened in the last 5 years coast-wide, that we have equipment, but we have to have folks to be able to go out and use that equipment. Dr. Raverty's been amazing with necropsy training around the province, going out and showing folks basic necropsy sampling. And I also do large whale satellite tag attachment training, which is learning how to attach satellite tags to the trailing gear of a mobile whale. We have a huge coastline, so it's really important that we have that capability. It often takes a day or 2 to get there. And then marine mammal lives training, which is quite important as well. And we have a lot of kits all over the province. And of course, you have to know how to use them as a group and deploy them quickly. Just getting to our response numbers, this was a record year. We had 833 reports. This is just a quick breakdown. I won't get into it, but typically we get almost half between pinnipeds and cetaceans broken down, and it's all about human interactions broken into those categories. Specifically around large whale entanglements, we had 20 reported so far this year, and 12 were confirmed, and that's just a breakdown. What I want to do is just show you some video from some of the rescues, because this one was done in October, and it was a unique humpback that had a rope through the baleen and had a buoy on one side and the trap on the other side. And it was an interesting rescue because it had companion animals. There were 3 companion animals. This animal was reported over 2 days, and it just shows how the 1-800 number is so important. We had 7 separate calls. Initially, the animal was observed over on the Sunshine Coast side. We had a team that went out and tried to locate, and by the time we were out there, we had sightings off Nanaimo. So we redeployed to Nanaimo. We had 5 or 6 boats out. BC Ferry saw the animal by Departure Bay. Unfortunately, we weren't able to find the animal. And as we were heading home on the boat, the animal was sighted up by Texada Island. And then this is the next day when we were out. Whale watchers found the animal off Texada and called it in, and we were able to get out, and fisher officers put a satellite tag on the animal, which is huge, and then we were able to work together. And you can see we put a working line on, and the animal did kind of a spy hop and a backflip. And the pressure of the buoys that we had on actually pulled out the rope without damaging the baleen, which is exactly what we want. The animal on the far right is the animal. This is him after he took off there. And everything worked out well, and that's what we want. The next one is kind of a, what's so funny? So this one is a gillnet, and this is a gray whale that it got caught up in a First Nation FSC Chinook fishery off Yuquat, which is near Nootka Sound. And it was they got caught up in the middle of the night, so it was quite dangerous. And we weren't able to locate this animal for a couple days. We got a satellite tag on it. We were able to get half the gear off, but due to weather and darkness, we had to put the sat tag back on. The animal went offshore about 40 nautical miles, and there was 35 knot winds for a couple days. This is when the animal came back in about 3 days later into Brooks Peninsula. So we had a flat, calm day. We were able to locate it because of the satellite tag on it, and then we worked up the gear, and you can see gillnet is very difficult to get off, very dangerous. We basically had to cut off three lead lines, 3 cork lines on the left fluke. And you can see this is after several hours. The animal actually just twisted out. We use drones extensively now to, first of all, identify the gear configuration, how it's entangled. And often it's 1 or 2 cuts. And if you know how it's entangled, and then also the drone is a safety issue if the animals come back towards us. And you can see also really important, before we used to guess that we got all the gear, this is the animal that took off and he was gear free. So it's a huge advance for us, not only safety for us, but also for the whale. I just wanted to, we're having a really busy year for dead marine mammals. Just in the last 5 weeks, we've had 5 dead humpbacks, with one, the latest one just announced yesterday afternoon off Ucluelet, 12 nautical miles off. Our DFO plane was able to photograph it. We haven't been able to secure that animal, but it's one that we're hoping to get to shore. We couldn't do this without Dr. Raverty. He's an amazing individual, and just I want to show this video of him, who is incredible. This is a humpback that died off Langara this year, and it was complicated because it was in a crevice, and again, he goes to lengths. He's on top of the whale. We were able to get a very isolated spot, very rough, and again, we used drones to film this, which was helpful. And of course, this was very unique. I hope nobody's had a big lunch there. And again, just how important it is to get there and find out if there's any anthropogenic or human cause of death. You'll see here, we actually had to get on top of the whale to get in there. And of course, Dr. Raverty's about 8'10", and I'm 5' nothing. If I fell in, I was definitely going to die, but he would have been okay. But just to show you how complicated some of these necropsy efforts are, and again, without the dedication to get out, get the samples. And I just, this is almost over, want to just, we'll just go through this, yeah. Sorry, it's a bit gory, but again, it's, I think we've done, in terms of necropsies, large whale necropsies, we're up to 8 this year. And again, this is the animal that was photographed yesterday afternoon, which looks fairly fresh, and of course that's the tongue there, which often happens, and one that we'll hopefully be able to secure and maybe look to do a necropsy in the next week. We also have an entangled humpback that we're trying to locate off Quatsino, which is another animal that's currently on our radar. So I think that's all I had, and yeah, if there's any questions.
Okay, thank you, Paul. We do have questions. I guess we'll start here, and then we'll go to Dom.
On that first map that you showed, did you guys get IDs on who that animal was?
Repeat the question.
On the first animal that was on there, did we get IDs?
Yeah, the one that had the line going through the baby.
Yes, yeah, we work with all the researchers. There's amazing humpback catalogs, as you know, and we were able to ID that animal, and you're going to ask me who that is, and I can't remember off, but we know who that animal is, and yes, we'll be keeping track of that animal as well. Yeah, sorry, I can't remember.
Yeah, I noted there were 130 killer whale reports. Can you tell us about any of those?
Yeah, so Dom's asking, he noted that there were 130 killer whale reports. The vast majority are disturbance reports, so perceived too close within the 100 meters. Thank you.
And as he's taking that off, and any insights into what's going on with the humpbacks, these recent deaths? Is it something to be alarmed about? Or is it being seen also in Washington State and Southeast Alaska?
Yeah, it seems like a blip, and as I say, it's 5 in 5 weeks or so. We've had 2 of those 5 that we've had blunt force trauma evidence, so consistent with a vessel strike, and the 2 others that were sampled, we're still waiting on samples, but one of them was very decomposed, so it's probably going to be difficult. But it doesn't, my colleagues haven't noted a pulse in this same time frame. So yeah, we're hoping that this is the last one for sure.
Okay. Thanks very much, Paul. And along those same lines, we have also a talk submitted by Wendy Slanislow. and on a very relevant topic.
I'll be speaking on marine mammal entanglement risk in BC, on-demand gear as a tool to reduce entanglement risk to large whales. The Canadian Wildlife Federation is undertaking entanglement mitigation work in Atlantic Canada to help minimize entanglement risk to the critically endangered North Atlantic right whales. The goal is to expand this work to the West Coast, and the work includes risk assessments and ropeless gear trials. Entanglement refers to the wrapping of fishing lines, netting, or marine debris around a whale. It can cause serious injuries to whales and affect their ability to swim, feed, and potentially result in starvation or drowning. To harvesters, whale entanglement may result in gear damage and gear loss. Humpback and gray whales are the most commonly entangled species of whales. Entanglement is not extensively studied. An initial pilot study by the Marine Education and Research Society in collaboration with DFO found that 47% of humpbacks have scarring on their tail stalks, indicating that they have been entangled and have survived. The DFO Cetacean Research Program has a record of 11 entanglements in BC crab, commercial, and prawn gear. The Strawberry Isle Marine Research Society has documented 16 gray whale and 11 humpback whale entanglements in Clayoquot Sound alone between 1995 and 2012. The entanglement risk assessment aims to look at the co-occurrence between commercial crab and prawn fisheries in BC and the abundance and distribution of select whale species. In this example, we're looking at commercial crab fishing effort and the abundance and distribution of gray whales in Barkley and Clayoquot Sound. CWF is working with lobster and snow crab fishermen to assess on-demand gear in the Maritimes. Fishermen have incentive to do this. In the Atlantic, there are fishery closures in place to minimize whale entanglements. Using ropeless gear allows them to fish. Here in the Pacific, there are no fishery closures to minimize whale entanglement. Other benefits to using the gear include potential reduction in gear theft and gear loss. Ropeless on-demand gear technology uses an acoustic signal to summon gear to the surface. To date, CWF has trialed 7 different acoustic systems with nearly 600 deployments in the Maritimes. We're trialing 2 systems here in BC and have 24 deployments to date. CWF has supported 10 fish harvesters with on-demand gear provided free of charge through its canned fish lending program this year. CWF has also provided reviews to gear developers on the fishery suitability of their gear to help improve their systems. All this feedback is directly from the harvesters. The system CWF is trialing is called the EdgeTech. The EdgeTech consists of what looks like a traditional lobster trap, and it has the end line coiled inside it. The end line is attached to the top of the EdgeTech system, which is acoustically released and floats to the surface. The ropeless riser system is truly a ropeless system in that it doesn't have an end line associated with it. It consists of a metal frame, a lift bag, and a canister of compressed air. You know where all your traps and your units are if you don't have traditional end lines with floats. The EdgeTech system has a trap tracker app, which allows you to mark and see exactly where all your units and traps are deployed. It's integrated with Navionics, so you could see this visually on a chart. Trialed the EdgeTech system with a prawn harvester in Alberni Inlet this fall and got it to a depth of 620 feet, which is a depth record for this system. We also tried the ropeless riser system and set another record at 720 feet. We had success trialing the EdgeTech in Tofino with a crab harvester who fished single pots. We chartered a commercial fishing guide out of Ucluelet to test both systems. Both EdgeTech and ropeless riser were successful, but we found it to be heavy and large for a small sport boat. We conducted 2 dock demos to date. And we have more dock demos and gear trials planned, continued engagement with fish harvesters, and data collection for the risk analysis. Thank you.
Okay, and we thank Wendy for that. Yeah, it's gear that's being used more and more on the East Coast. Initially, there was a lot of reluctance from the fisheries to adopt it, and now there's greater buy-in because without it, they're not going to be able to fish. And they seem to be making it work quite well from the reports that I've been seeing. I'm going to wrap things up here with a talk to give. I don't know how many of you are aware of Bill C-251, which is called the short term that the government of Canada was using was the Conservation of Fish Stocks and Management of Pinnipeds Act. I've heard many people refer to it as the Seal Slaughter Act. And it's legislation which I just want to bring you up to date on what's happened to it, some background, and have a quick discussion on it. So the official act, and you can go on to the website, the Parliament of Canada, and the official name was an act respecting the development of a federal framework on the conservation of fish stocks and management of pinnipeds. DFO had no part in this. It, I think, surprised people in DFO as much as it surprised non-DFO people that this was going through the House of Commons. It's a private member's bill. It was sponsored by Clifford Small, who is a Conservative member of Parliament from Newfoundland. And you can go onto the website and you can find that getting new legislation passed, it's a very involved process. The House of Commons has a first reading, a second reading, then it goes to committee, then there's a report, and then there's a third reading. And if it gets through that, then it goes to the Senate, to a first reading, a second reading, and then a third reading, and then it becomes law. And you can follow through the process and see what's being said. You can also click in this box to get the text to see, well, what is this new law going to be? And on here, I'm just going to give you a few excerpts because I don't want to read out everything. It's got 7 different measures, and I'm just going to talk about 2 here. It did everything from trying to create a bigger market, helping support it, doing annual census counts. But the 2 here that caught my attention in particular was A, to promote the conservation and protection of marine ecosystems by addressing the harms caused by pinniped overpopulation. Of course, that's a value-laden term, and one could debate what does that mean. The other one is Clause E, is if it passes, it was to promote year-round control of pinnipeds. Now, people say that's not culling, but in fact, that's what it is. In order to manage their numbers and mitigate the detrimental effect of these marine mammals are having on the environment when acceptable pinniped population levels are exceeded. So on February 9th of this year, Mr. Small stood up and just made a few short sentences about the introduction of his bill for the first reading. Such all that happens at a first reading is just tabled. And 2 things I've extracted from what he said. He says, conservation efforts by commercial and recreational fisheries have been ineffective in the rebuilding of fish stocks, and we are now faced with an ecological disaster. Now, there's no human-related part to this ecological disaster. It's clear what the problem is. And it goes on, and sorry it's a little bit long-winded, but it says, pinnipeds, a group of marine animals that include seals and sea lions, have populations that are now 2 to 5 times higher than historical levels. Of course, if we want to define historical levels, referring after they were culled and reduced. So it all depends on your sense of history. In coastal Canada, they consume 50 times more fish than our fishers harvest. I think we need to look at what year we're referring to. Should this bill pass, the Minister of Fisheries and Oceans would create a framework to manage pinnipeds through consultation with Indigenous groups and other industry stakeholders, as well as Canadian international scientists to, and I've underlined this, to restore balance to our marine ecosystems. And so the perception in amongst many politicians, and of course many people in BC too, both East and West Coast, is that our ecosystems are out of balance and something has to be done because seals and sea lions have caused this problem. So it's put in, then the second reading comes up, and it starts first on April the 28th with 4 major speeches that are made. And you can get copies, everything is recorded. There was a speech from Mr. Small, who tabled the bill, a speech from the Bloc Québécois, a speech from the Liberals, and a speech from the NDP. And what's interesting is that none of them speak against the bill, but two, in fact, didn't speak in favor of it either. And I guess this is probably politics, that you don't really want to speak too strongly, you need to be sympathetic, because certainly there are many fishers who are very concerned about their livelihoods. Things aren't good, it is a tough livelihood, and when things aren't going well, often somebody has to be blamed, and in many cases now, I think it is seals and sea lions. So on this, in fact, it was the Liberals and NDP that did not speak in favor of it, but nor did they speak against it. So June 15th, the vote comes, and you might be wondering, how did that vote go? It's in parliamentary language, so 6 are paired. That means that they abstained. Yeas, 150 supporting it. Nays, 175. And I haven't gone through to figure out who voted how, but all the names are there. I just don't know the parties, but my quick read of it is that those in favor of the, I'm going to call it the seal cull or the act here, are Conservative members of Parliament and the Bloc Québécois. Those opposed were the Liberals and NDP, although I haven't gone through to check that out. But it does tell me, so it's failed. It has stopped at this point. But it just takes a change in government, and it's going to be back on the table. So if you go onto the site, you'll find here, shown as the bill was defeated, a second reading, and so that's it for now. But just like you think, you know, have the seals dodged a bullet? Well, the reality is that there's a huge amount of this rhetoric and misinformation is coming from British Columbia. It's here. And I think it needs to be addressed. We've been quiet about it. There was just recently a SEAL summit in Newfoundland, dealing with people from industry, focused quite heavily on people concerned with marketing and selling seals and sea lions here for restaurant food, for selling the furs and whatnot. So I think it does need to be addressed. I'd be very interested in talking to people that are also concerned about this. I think it's part in working with both, yes, we need to understand where the concerns are coming from, but there's a huge amount of research being done as well. I think that can show the role that they are playing and put things into perspective, and pointing out, for example, that I'm not sure that we need people to balance the ecosystem. I think it's kind of preposterous. I think, in fact, the transient killer whales are doing an amazing job all on their own of bringing back an ecosystem to make it healthier, more sustainable, with greater productivity. Thank you very much. Anyway, as I said, I'd be very happy to talk to others about continuing this and seeing what can be done to provide some balance into the discussion. I think that is bringing us to the end. Here's this point, I ask if anybody has anything they were hoping to come talk about or say. This is your last chance. If not, I would say that many of us are going to be hanging around in this room and outside. We also have a couple of pubs on campus. There's Brown's just over here, and I'm sure, I know I would love a beer at this point. But any of you that like to get together and talk longer, we'd be happy to do that. So just in wrapping up, I do want to acknowledge people. We came close to thinking we were ready to be back in person. I'm so glad we did it. You know, I think all of us need this to see one another again. And I'm very pleased. I hope next year we can boost our numbers or allow more into the room. I think we're getting back to there. I appreciate those that were watching today online and stayed in with us. We'd like to have you in the room here. I know Marty Helena particularly wants to see you here as well. So anyway, we look forward to that. But I really want to put a shout out to Marcus, who without him, I probably would have said maybe it's too soon, but Marcus said, no, we're doing it. So thank you, Marcus. Thank you, Christine at the back, who's always here too to help make things run, the silent partner. Also want to thank Rob
who just helps us brainstorm this and get through it. So it makes my job much easier. Outside, I think most of you, or if not outside, didn't see, but on email you were dealing with Pamela Rosenbaum, and she's helped make sure that there was food, took care of this, registrations. I'm very indebted to her too. The postdocs in my lab and Dave's lab and our grad students, they too make sure that this event happens. So to all those people, I am very grateful. We will be talking about ways to get a committee, because this has been 30 years that I've been doing this. I can't do it for another 30. I would love to think I could, but Marcus and I and Dave will be talking about ways in which to include more people. Finally, I want to thank our sponsors, the Pacific Whale Watch Association. They've been there for us from the beginning. That's why you get food. That's what helps make this happen. The Porpoise Conservation Society, we can thank them for our technology and the work that Anna Hall and Marcus and others do for them. That makes it. The Pacific Wildlife Foundation, which a number of us are part of, we've always been a count on them as well. And I know they've been online today watching this. And finally, UBC and the Institute for the Oceans and Fisheries for giving us this space and time here at UBC. And last but not least, all of you for continuing to come, and all of our speakers. The talks were awesome. I so enjoyed it. It was just a great day. I look forward to it next year. Thank you, everybody.